an honor to be a speaker in this conference. And the work that I'm going to present is, of course, a contribution of many, many colleagues and students. So let's see. As a background, I would like to position my work, our work, in what is called tertiary data analysis. This slide is taken from a spin-off company of Mike Stonebreaker, which is the current Turing Award, and essentially showing that in genomics, and in particular in data management for genomics, there is a growing interest not only in primary and secondary analysis, which have to do with essentially calling mutation, calling peaks of expression and so on, but also in tertiary data analysis. That is to say, how to make sense of the data, of the signals which come from the genome. And this is the motivation of the title of my talk. I think there was something. Okay, something is happening. Let me go back. Actually, I would like to go back to, okay. Sorry that I have been going too fast. All right, so I just skipped one slide, which actually had to do with a short history of genomic computing in our department. We started with essentially a meeting where I was introduced to this topic, which only dates 4 years ago. We have now a relatively large group. We had this project that was mentioned by Marco, the PRIN project, the first release of our system in 2015, the Advantage Graphs, which has been mentioned, and the second release, which is currently available at Cineca, which is a major computing facility in Italy. As I said, this is the background, and by interacting a lot with the biologist of Yale, the Institute for Oncology, European Institute for Oncology. Essentially, we came up to understand that their main questions were the following. The first one is, can we characterize which are the interesting regions to be discovered with the genome-wide query? And this we did essentially by inventing what we call GMQL. It's a new language for querying the genome, which is essentially the main contribution of this speech. But then we think that it is also possible to, for instance, cluster and, let's say, genomic data of patients according to clinical phenotype and compare them. And again, the idea is to have first an extraction by means of queries and then clustering applied to the result. And similarly, we could use data analysis on top of the result of a query to answer questions such as extracting all the features of genes involved in the same biological process. And I got my first ERC in search. One of the main, let's say, objective of my research is to be able to search the genomic data. And of course, once we have extracted the data, you can apply indexing and search. So the idea is to have a core genomic query language in the middle, and then all these kind of technologies around. The data model that we propose is relatively simple. It is based on regions and metadata. The regions are essentially, let's say, characterized by their coordinates, chromosome stop strand, and then any data type. So in this particular example, the data type is quite simple. It is just an information about, for instance, the significance of a peak value, but it can be an arbitrary type. The metadata, after exploring the variety of possibilities of, let's say, the word of description of metadata, we ended up with a solution which is the most simple possible one, which is to characterize metadata by attribute value type in a semi-structured format. So this accommodates any particular kinds of metadata. This is an example of a data set which has 3 samples. The samples are a combination of regions and metadata. the metadata describe three particular patients with their tumor type, which is BRCA, and additional information, which can be of any type. And the idea is that by means of this genetic format, we can accommodate any, let's say, format which exists which has been produced by next-generation sequencing, including mutation, expression data, and all the variety of types which have been discussed also in the previous presentation. And the query language. The query language is a high-level language, so the idea is to rise up the level of abstraction compared to the current let's say, languages which are available in the bioinformatics community. It is inspired by a language which is called piglating, which was presented in the world of Hadoop. And it includes several high-level operations, some of which are classical in the relational terminology, such as selection, projection, grouping, ordering, union, difference, and merge. Some of them have been specifically invented for the domain of genomics. And I will give you a very simple example of what we mean by the query language by this very, very simple query, which essentially is selecting the promoters from annotations, is selecting the ChIP-seq experiments from ENCODE, and is mapping the peaks of every ChIP-seq peak over all the promoters and counting how many peaks are there. So this can be done in 3 lines of code. There is implicit iteration, there is implicit, say, access to the data. The result brings about the metadata from the initial data sets. So essentially, you can see that, for instance, I projected some of the metadata of the first 3, let's say, by ordering by the count of peaks. And the idea is that we can therefore execute this operation very fast on top of processed data. The diagram below shows that our main operation, the one that have been developed for genomics, are join, map, and count. And we show that the performance for doing join, map, and count over thousands of samples and millions of regions is relatively acceptable. So the idea that we like to apply this language to process data of any kind, which are currently available in large data sets such as ENCODE, TCGA, and so on. I will just show you one of the operations. There are 10 operations, but one of them is a representative, just to give you a flavor of the language. So we can, for instance, map. That means count the regions which how many regions of what we call the experiment intersect with regions which we call references. So in this case, the references are 2 genes. The experiments are mutation. By mapping, essentially what you do is to extract all the mutations which intersect with the particular genes, and then you count. And then, so the map operation will produce the counter of how many mutations appear within the region of the genes. By doing this, we have what we call an abstraction, which we consider to be very relevant, which is called genome space. So you understand that we can query arbitrary types of information. Here there is an example where we essentially explain, well, visually reproduce several kinds of data types. And we can now try take some operation which essentially extracts or maps this information in a very simple format where we have a region on one side and experiment on the other side. And this simple format is really nothing more than an Excel table. Of course, instead of extracting a single value, you could extract a feature vector, something more complex than that. And then this can be represented as a heat map, and therefore you can have a visualization of this genome space. Or you could also interpret it as a network, and therefore you can do network analysis on top of this result. And this is, for instance, one of the classical networks that can be extracted as a result of a query. We are working also on collecting data, so we have integrated process data, not raw data, process data from ENCODE, epigenetic, roadmap, and TCGA. These are the, let's say, the number of samples and file size. We are not talking about really big data here, but we are talking about whatever has been produced by this community of scientists and put available, as Serafin said in the previous talk, for making additional, let's say, findings. The architecture is, I will go very quickly to the architecture because I understand you are not computer science addicts, as I am. The architecture is relatively neat because we have essentially an intermediate representation from which we generate our, which is generated from our GMQL language. And we have actually supporting 3 major implementations which are using Spark, Flink, and SciDB. Spark and Flink are 2 engines which are very well known in the cloud computing community. SciDB is a specific vertical, specific database system which has been designed for scientific application by this Mike Breaker company. So the system has a lot of components from the top level where we expose a web interface to the bottom level where we expose 3 implementations. In between we have a lot of layers. It has been engineered in a relatively solid way. And it gives a lot of thinking also from a pure computer science point of view because there are options for optimization. When we map to an information language, we can do better exploitation of, let's say, the operations. And, of course, we can do exploitation of the underlying engine by doing low-level optimization. I will not insist very much on this, but there are many interesting problems about how you bin the genome into pieces. And then you can partition the piece, and then you can use each partition so that you can produce cloud-based computation, which are performed using Flink and Spark. In my community, we've been using GMQL as a sort of framework where we could compare the performances of these systems. For instance, this is a comparison between Flink and Spark at work. And you can imagine that there is a optimal binning size, which is a size at which the operation on the genomes are better parallelized. Of course, if you have too fine or too coarse granularity of the bins, you have worse performance. I will move to some description of some of the biological work that has been possible through using the system. In particular, I will focus on one. We have been looking at the recent paper, which was published on Nature, which has to do with how topological domains of the genome can influence, in particular, can motivate, and can be one of the causes of cancer. In particular, this paper by Flanagan has shown that a particular kind of cancer, IDH mutant glioma, is explained by the fact that there are some boundaries between TADs which get destroyed, and therefore some genes which normally do not interact because they belong to different topological domains come in connection, and in particular it showed that there are oncogenes which are specifically related to IDH mutated glioma are let's say, not regulated in this particular disease. So we have been, first of all, I show you this as an example of how compact is the program that would essentially compute the genes which belongs to the to same TAD as opposed to genes which belongs to which are one TAD apart. Because the idea is that there is a higher correlation between genes which belong to the same domain, and lower correlation between genes which are separated by one of these TADs boundary. So what we did was first to replicate the result of Flanagan. This is one let's say, figure taken from their paper, where we show that for the particular kind of tissue that they have been selecting, the correlation of the same domain is higher than the correlation of different domain. But of course, we could then use this exact program and change the tissue, and therefore do the same exact evaluation for different tissues. And we did it for breast cancer, where the, let's say, this phenomena is more visible. We did it for other tissues, such as skin, lung, adipose, and artery. We also tried to put tissues of humans into the TADs of mouse. This was just more for, as a, let's say, a divertissement, we would say, because there is but it shows that the TADs are replicated across the various species. And we can show diagrams such as this one, where we show for each TADs and for each tissue how many, let's say, active genes are there. So showing, this would be the result of a map in terms of our operation. And we are working with biologists on many other projects. We are working on, let's say, the microbiome, in particular for a specific disease. And we are looking for dimers, particular pairs of transcription factors that co-regulate genes. We are looking on particular transcription factor co-occurring with particular binding sites, and also looking for areas which are dense of transcription factors. So these are examples of the kinds of problems that can be studied by using typically the data, in most cases public data, and using the system. Our vision. The idea is that we are confronting a world where there are a lot of excellent programs for doing what we call secondary analysis. Let's say DNA reading, alignment, calling of peaks, calling of mutation, and so on. And we think that also along the dimension that we have been discussing this morning in Seraphine's talk, that we are going toward a world where there will be a lot more of process data available, and therefore tertiary analysis will become the more important and more distinctive part to be studied. We want to essentially improve our system by doing several things, such as tracing metadata, extracting by means of patterns, regions, and this will be the topic of the next speech. Doing the statistics, also integrating with interaction networks, also making sure that integrated repositories with data from TCGA ENCODE and 1,000 genomes and roadmap of GenoMetric and possibly many other talk the same language, so within the same context, with the same approach to metadata, publishing web services where people could use GMQL or GMQL-based query services. And the most interesting idea along the vision that was presented today would be to provide a basic technology which could allow an Internet of Genome, that is to say, a possibility for genomic data to be accessed and retrieved. So if we go in the direction of making the genome more popular and more user available, then this would be an enabling technology, and also enabling metadata and feature-based search, sort of searching in the same way as we have been learning through Google. So as a conclusion, we have, I think, an interesting project. We have been also able to put out several collaborations, two of which will be presented in the next presentations. We have collaboration not only within the computer science community, but also within the data science community, and with biological collaborations. And there are resources, and in particular, one resource is our website, which is open at the Cineca, available for use. This is the end of my presentation.